Humans consume massive, complex structures every day: structural proteins, starches, and triglycerides. In their raw form, these macromolecules are entirely unusable by ourselves. The body faces a central biological tension. It must deploy destructive forces to tear these structures down into microscopic building blocks, without allowing those same forces to digest its own tissues. This gauntlet begins in the mouth. Through mastication, the physical crushing of food breaks the mass into smaller pieces, which drastically increases the surface area exposed to chemical attack. During this physical breakdown, the salivary glands release the first chemical strike, a specialized enzyme called salivary alpha amylase that begins targeting carbohydrates. Alpha amylase targets dietary starch by randomly severing its internal alpha-1,4 chemical bonds, reducing the long chains into smaller fragments called dextrins. The mouth acts as a preparation phase, transforming food into a chemically vulnerable mass called a bolus, ready for more aggressive dismantling. The bolus is driven down the esophagus by peristalsis, rhythmic, involuntary muscular waves that propel the contents into the stomach. The stomach environment is extremely hostile, featuring a dramatic top in pH to a highly acidic level of 2. This acidity causes immediate collateral damage to the initial digestive process, as it instantly neutralizes and halts the action of the salivary alpha amylase. The acid's primary objective is to denature dietary proteins, forcefully unfolding their complex 3D structures into exposed linear chains. To protect itself from its own secretions, the stomach lines its walls with mucus and employs a zymogen strategy, secreting only the inactive precursor enzyme pepsinogen. Contact with the hydrochloric acid triggers the activation of pepsinogen into its active form, pepsin, which begins slicing the unfolded protein chains into smaller peptides. This chemical violence reduces the meal to a highly acidic, partially digested sludge called chyme. As the acidic chyme enters the duodenum, it triggers the release of 2 vital hormones, cholecystokinin and secretin. These hormones signal the pancreas to deploy a payload of bicarbonate and a cocktail of inactive zymogens directly into the intestinal lumen. Digestion here is ignited by enteropeptidase, an enzyme on the intestinal wall that cleaves trypsinogen into the active enzyme trypsin. Trypsin initiates an autocatalytic cascade, rapidly activating all other pancreatic proteases in a massive chain reaction. This diagram shows one branch of that activation. Trypsin converts inactive chymotrypsinogen into pi-chymotrypsin. The molecule then cleaves itself further to become fully active alpha-chymotrypsin, finally ready to dismantle proteins. This delayed activation allows the body to deploy these weapons only where food is present, leaving the pancreas itself protected. Dietary fats present a physical challenge because they are water-insoluble and clump together, resisting the action of enzymes. To solve this, the liver and gallbladder deploy bile salts into the duodenum to act as chemical detergents. Through emulsification, the bile salts physically smash large fat droplets apart, drastically increasing the surface area available for enzymatic attack. Pancreatic lipase, assisted by colipase, can then strip the fatty acids away from the triglyceride cores. Carbohydrate digestion also resumes in the small intestine. This chart shows pancreatic alpha amylase continuing the breakdown of dextrins into maltose. The final breakdown occurs at the intestinal wall, where brush border enzymes like sucrase and lactase create single monosaccharides like glucose and fructose. At this stage, carbohydrates, proteins, and fats have been systematically dismantled into their fundamental molecular building blocks. Deconstruction is meaningless without absorption. To see how these nutrients enter the body, we must examine the microscopic anatomy of the intestinal wall. The interior of the small intestine features large folds covered in finger-like projections called villi, which are themselves blanketed in microscopic microvilli. This structure multiplies the absorptive capacity 600 times, packing an area roughly the size of a tennis court inside the gut. Water-soluble nutrients, such as amino acids and glucose, cross the cell barrier through diffusion and specialized transporters to enter the bloodstream directly. Fat-soluble nutrients follow a more complex route, ferried to the cell border inside tiny water-soluble spheres of bile salts called micelles. Once inside the enterocyte cell, The fat pieces are rebuilt into triglycerides and coated in a protein shell to form a chylomicron. These chylomicrons are too large for blood capillaries, so they are expelled from the cell and enter the lymphatic vessels instead. From the initial mechanical crushing in the mouth to the microscopic transport across the intestinal wall, this digestive sequence extracts the molecular building blocks required to sustain human life.